Hello, I'm Tom Luchtefeld, and today I'm going to be talking to you about my third rotation at Johns Hopkins School of Public Health in Dr. Jimin Wong's laboratory. In this lab, I worked on chromatin immunoprecipitation sequencing data. I used clustering algorithms and natural language processing. Let's go ahead and get started. So just a quick review of what chromatin immunoprecipitation is. CHIP can be used to figure out where proteins of interest attach to DNA. In this example, p53 is the protein of interest. An antibody is used that is specific for p53, and then the DNA is sonicated. We can use this process to purify DNA fragments corresponding to where p53 attaches to DNA. We can then amplify these fragments and use sequencing to figure out what these fragments actually are, and then we can use the Burrows-Wheeler transform to figure out where these fragments map to the human genome. I use the Chip-Seq application, which is written in Java and is open source, to do all my visualization and analysis. I also wrote my own code for implementing natural language processing and some clustering algorithms, which we'll get into in a bit. This data set that I worked with was RNA polymerase II and various modifications of RNA polymerase II. Just go over the standard transcription theory real quick. We won't go into the details. Essentially, RNA polymerase II goes under different phosphorylations during transcription. There's a serine 5 phosphorylation, which helps to recruit the cap, and then there's a serine 2 phosphorylation, which initiates elongation. This process is reversed when RNA polymerase II detaches from DNA. Taking a quick look at the distribution of reads on genes, we can see that for the majority of genes there are no reads, and if we ignore all the genes for which there are no reads, we get a log normal distribution of reads. And we see that for the most part we have around 300 reads on genes, and that's probably the most probable. That's around the expected number of reads per gene among the non-zero reads genes. We can visualize where these reads lie on each of the 24 chromosomes, and there's no mitochondrial chromosome in this data. So does the model fit the standard transcription theory? Well, to figure that out, we can take an average gene and we can look at the transcriptional start site, which here would be 500 base pairs in. So what we see here is that serine 5 is typically more concentrated before elongation begins, and serine 2 is less present before transcription begins, before elongation begins. This fits with the standard transcription theory, and serine 7 phosphorylation doesn't quite fit, but it's possible that we would see this kind of distribution because we see that it's most highly concentrated before transcription starts, but also gradually falls until about 2,000 base pairs out. This is also not normalized for genome length, so we could have very short for gene length. So we could have very short genes that are going to be affecting the way this distributes. If we look at across the entire gene, and this is a normalized distance across a gene probe, we see that in general serine 5 and serine 7 phosphorylation in this Chip-Seq data are highly concentrated at the 3' side, and serine 2 phosphorylation is more level across the entire gene for an average gene. So what can we do with this kind of data? Well, something we might want to know is alternative modes of regulation for our RNA polymerase. To do that, I was thinking that I could do some clustering on this data. Because this was an abbreviated 4-week rotation period, I just used a simple clustering algorithm. I used k-means. K-means works by allowing the user to guess at the number of clusters they expect to see a priori. In this case, looking at the data, we would guess that there are 2 clusters, and so we can put down 2 centroids. Now we count each of the data points and we find their distance to each of the centroids, and we update the centroid to the centroid of the data points. So we see here that we have a red X, and we find all the points closest to the red X. We then update the location of the red X to the centroid of all those points. And we do the same for the blue X. After several iterations, you can see that the data points have been put into their correct groups. To work with continuous or pseudo-continuous data, like our gene probes, we need to discretize it. So what? To do this, we can break our probes up into bins, and we can find the average number of reads per bin. In my analysis, I used 10 bins, and I tried 20 bins as well. It didn't seem to make a whole lot difference. Doing this kind of process is going to end up with creating a fairly large dimensional vector, and k-means is going to suffer a lot once you get much past 10 dimensions. So we look at serine-2 phosphorylation, and we say a priori that we expect there to be 3 groups. We can start the iteration process, and we can see that one of these clusters after 2 iterations looks significantly different from the other 2, with a large amount of serine-2 phosphorylation peaking near the 5' side, and with the other group having somewhat of the standard serine-2 distribution. We can see after 5 iterations that this process has become even more divergent, and we see that for 2 of the clusters they're pretty similar, but for this cluster 0, which involves 5,200 or 1,389 genes, so the fewest number of genes, we see that serine-2 phosphorylation is most concentrated near the 5' side, the terminal side. After 20 more iterations, we see that the clusters aren't changing a whole lot, and that the distribution across the probe is relatively standard. So how does this apply to some actual models of transcription? Well, let's take a look at transcriptional termination. There are 2 models for transcriptional termination. And although neither have been completely confirmed, we can still look at them and see what our clustering data says about them. In the allosteric model, RNA polymerase II transcribes through the poly A tail, and then eventually it just kind of falls off the RNA, becomes less processive and stable after transcribing through this tail. In the torpedo model, after transcribing through the poly A tail, we see that a 5' to 3' exonuclease binds to the RNA transcript and then basically torpedoes the RNA polymerase II off. If we were to hypothesize how the distributions across the gene probe would look for these different models, we might say that in the allosteric model, we would expect RNA polymerase II, and we can say the serine-2 phosphorylation, to be more gradually falling off of the DNA, whereas in the torpedo model, we might expect a sudden fall off of the DNA. Doing this clustering process, what we end up seeing is 2 pretty well-defined clusters. And one of these clusters, we see that serine-2 phosphorylation is really peaking, and right before the transcriptional termination, which we've placed 500 base pairs into this probe. And we see that for the other cluster, that the distribution is fairly gradual across this probe, and we might expect it to reduce more if we had looked farther, more base pairs down. Another alternative method of transcriptional initiation or transcriptional regulation would be heat shock elements, and this involves polymerase pausing at the 3' side of transcription. So in this case, essentially there is a complex that holds RNA polymerase II in place. When the cell undergoes some stress, some state changes occur, and the RNA polymerase II is released to very quickly start transcribing. If we take a look at the complexes involved in this, there's a particularly interesting protein here called NLP, which is removed for the RNA polymerase II when it starts transcribing. So to model this process, we can measure the distribution over 500 base pairs upstream and downstream of the transcriptional start site. And because serine-5 is phosphorylated both in the poised polymerase and the productive elongation, we're going to look at that data set. What we end up finding is 11,000 cluster 0 genes and 5,000 cluster 1 genes. Let's look at how those clusters end up distributing across our probe. We see that in one of these clusters, presumably the polymerase poison cluster, we have a large peak for serine phosphorylation just slightly downstream of the transcriptional start site. It's exactly what we would expect to see for polymerase poison. For the other cluster, we see kind of a more standard distribution across the transcriptional start site of serine-5. Now we also know that we should expect to see the NLP complex distributed in the same way. We could have hypothesized before that the NLP complex should be highly concentrated near the transcriptional start site for genes in which polymerase poisoning was occurring. If we take a look at this data, yellow here, SPT5, is actually the NALF complex in humans. We can see that it does in fact follow this what we would expect this distribution to be quite closely. This is a nice corroboration of what we would think the model would be. So how do we actually analyze these clusters? We can know a little bit about molecular biology and genetics beforehand, and we can say, well, we expect these to be the models of transcriptional regulation, or we could try to learn what these genes actually have in common with each other. To do this, we would want to do some term importance studies. So when I was starting out with this project, and again, it was a short rotation, what I did was I downloaded the last 25 papers for every single gene. And there's a better way to do it than I ended up doing. I ended up using the names for each gene directly out of the HG19 reference, and there are better resources for finding PubMed articles for each gene, but it worked fairly well. I created a corpus of 1.1 billion words with 47,000 articles, and that was a large enough corpus, in my opinion, to start doing a little bit of word importance. So what does word importance mean? Essentially what we're looking at is if we have an article for a gene, we want to know what words are most descriptive of that article. The first thing we might think to do is just count how many times each word occurs. That gives us the frequency of the word in the document, the term frequency. Now, if that were all we were to use, we would find words like the and of to be incredibly important in describing every single gene. And so to fix that problem, we introduce inverse document frequency. An inverse document frequency, n is the total number of articles, which is 47,000 in our case, and the magnitude of dnn tells us how many articles our term occurs in. So a term like the occurs in every single article, and so the inverse document frequency would be 1. But a term like RNA polymerase might only occur in 1/10 of the articles, so the inverse document frequency would be 10, and it could even be higher. The importance then is just term frequency multiplied by inverse document frequency. For the word the term frequency is going to be quite high in any article, but the inverse document frequency is going to be very low. For a term like RNA polymerase, the term frequency is going to be very low in papers that aren't talking about transcription, but it could be quite high in a paper about RNA polymerase, whereas the inverse document frequency will remain high in both of those instances. So let's take an example of a gene that's commonly studied in my department. This is NQ01. NQ01 is involved with NRF2 and KEAP1 and the aryl hydrocarbon response. NQ01 deals with antioxidants and xenobiotics and metabolism. And if I were to pick words out that I would try to describe NQ01 with, these are pretty much the words I would pick. KEAP1 would definitely be up there near the top. KEAP1 targets NRF2 for degradation. Aryl hydrocarbon, this actually points out a weakness in the simplistic design of my code. Because we're only using single words and not pairs of words or tumors, we don't notice that this word aryl is probably occurring right before hydrocarbon in most of its occurrences. But we know that the NQ01 is involved with aryl hydrocarbons, antioxidants, xenobiotics, oxidative, and attenuation. I mean, I don't know what that is doing there, but we should expect these results to have some spurious words involved with them. So that's how it works for a single gene, but what about a cluster of genes? So to find word importance for a cluster of genes, we want to do the process on every single gene, and then we create a word importance vector. We create a word importance vector for each of those genes, and then we basically add them up and divide by the number of words. So we create a balanced word importance. Let's take an example for schizophrenia here. If we look at genes that we know are associated with schizophrenia, and I just found this from just a Google search, I looked up schizophrenia genes and I found this image. I can then go into my corpus and I can look up these 5 words, and I can run my balanced word importance algorithm. And when I end up getting out, there's a little bit of noise, but we end up seeing schizophrenia being the 5th most important word. So I think that's a pretty good result. Keep in mind that the algorithm had no idea that we were targeting genes for schizophrenia. It had no information about that. It just so happens that schizophrenia has a high enough term frequency and a high enough inverse document frequency that it ended up being a very important word for all of these genes, or at least a lot of them. We also see some other words which might have some interest. Oligodendrocyte, that's a brain cell, and then we have myelin, which is associated with oligodendrocytes. So, maybe we could look at these papers and realize that maybe part of the process of schizophrenia involves degradation of myelin on neurons or something like that. It's a possibility. We might expect that cocaine has a relationship with schizophrenia. There's some interesting patterns we could pull out of this data. So when I actually ran this on our alternatively regulated clusters, I got some questionable results. First of all, if we look at our serine-2, where we had this cluster that kind of got very high near the transcriptional termination site, we see that we get a small nuclear RNA, SN RNA, and we see RNA polymerase 3, which we would expect to be involved with U6, but then we see a bunch of words that maybe aren't involved. We know snRNA is going to be involved with U6. Short hairpin RNA, I guess that's siRNA. I don't really know that much about it, but we might expect it to be involved with alternative alternative transcription, and certainly the small nuclear RNAs are, particularly with splicing. But a lot of these terms don't really seem to fit, although we could look into a couple of them more closely. More troubling is that the same terms seem to apply to a lot of these alternative clusters. If we look at what we thought was alternative transcriptional initiation, what we end up seeing, alternative transcriptional termination, what we end up seeing is for what we thought was the polymerase, what we end up seeing is the same terms. We see U1 and we see splicing and polyadenylation, U4, U6. So it could be just that these snRNPs are involved in all kinds of alternative transcriptional regulation, or it could be that we're getting just the same terms for a lot of these genes. So because I was suspicious that the real cause of the terms we were getting was just that our clusters involved a lot of genes and that all of these genes are expressed in our cell model, I thought I'd look at some of these word importance vectors a little more closely. So using the clusters above, I thought one of the big problems would be just that we had such large clusters. One way to fix this problem would be to create a small set of strong example genes. In this case, I picked 100 example genes. First, I wanted to make sure that I was picking the right example genes. So if I take the average distribution of reads for my example genes and I compare their distribution to the cluster, I should expect that the distributions should look quite similar. I should also expect that for my smaller set of genes that we'd have a lot more noise, and that's exactly what we end up seeing here. When I use David, which is an ontology program which lets me look up terms associated with sets of genes, I don't really see any terms coming out, although possibly alternative splicing and polymorphism. But these enrichment scores are quite low. I'd say that these results are hard to have confidence in. If we take a look at the top 10 genes applying the same process, we see that a lot of these genes are small nucleolar RNAs, which possibly explains how a lot of the terms we got had to deal with small nucleolar RNA. If we look at the actual NLP report for these genes, we see all the snRNPs, which I've grouped up here at the top. We see the spliceosome showing up, cap bindings, short hairpin RNA, microRNA, and lupus actually shows up. And lupus has anti-U1 antibodies, autoantibodies. So we might expect that lupus involves dysregulation of alternative transcription or splicing. Just taking a look at the word importance density, we see that above 0.2, we get pretty rapid decay in word importance. Now, if we looked at our polymerase poisoning again and poisoning on the transcriptional start site, we could do the same sort of process. We once again see that our alternative transcription, if we pick the top 100 genes, the distribution follows the underlying cluster quite closely. You see orange is overlapping green quite well. The same actually happens for the other cluster, but we're going to focus on the alternative transcription case, which is polymerase poisoning. If we take a look at David, we end up getting a lot of terms which I would say would be hard to trust, but more importantly, if we look at the terms for cluster 0 and compare them to the terms for cluster 1, we end up seeing the same terms. What this means, my hypothesis is that essentially what we're finding is that we're finding all the genes that are transcribed in this cell model. And those genes may have something in common with each other, so that even if you separate genes into separate clusters like this, David is going to find the overall most descriptive terms, and it's just going to be terms that apply to all of these genes rather than genes within a subset. So we want to know what the difference between these two clusters is. We can do a projection, and we can subtract off the projection of one word importance vector from another word importance vector. So if we have these two word importance vectors, A and B, we can say take one word importance vector and subtract off the projection of it onto the other word importance vector. And you can see a visualization of that here. If we do this, we ended up getting quite nice results, exactly as we would expect. The word importance, the words that have high importance for both or low importance for both end up having values close to 0. You can see the mean is quite close to 0 for this middle peak here. Words that describe one cluster but not the other might be negative or positive, but we should expect to see this 3 peak density. And it's nice that this ends up being the results. So if we look at the results for this for polymerase poisoning, we end up getting, well, I was really hoping we would see something involving heat shock factor, but we didn't get that. If you had gone down far enough, you would have, but I didn't want to change the results for this case. We end up getting some SNRNPs again, but we get some other interesting things as well. The polyadenylation complex, this was a little surprising here because we're talking about transcriptional start sites now, and the polyadenylation tail of the DNA, this is near the 5' side, not the 3' side. PDCD4, I'm going to talk about a couple of these right now. Oh, I'm sorry. So it turns out that U1 associates with regulating transcriptional initiation. You know, going in here and looking up these terms and looking up with their relationship to transcription is a little bit is cheating a little bit, because we can probably find a lot of associations between different proteins and transcription. But it's nice to see that we have one paper that at least directly says that U1 is associated with TF2H and regulates transcriptional initiation. PAF1 is apparently implicated in transcription initiation, according to another source, and it's currently characterized for roles during transcription and elongation. Now, it's interesting, I found another paper talking about promoter proximal polyadenylation, and this explains how polyadenylation probably got so high, because when we see promoter proximal polyadenylation, we see sites with a reduced transcriptional activity. It could be that this is actually polymerase poisoning, or at least related to it. And we see promoter proximal is actually a pretty high word importance term, and polyadenylation is as well. So if we had used tumors, as I was discussing earlier, where we have pairs of words rather than single words, we might have seen these two terms put right next to each other. Of course, splicing is functionally coupled with transcriptional elongation and initiation and termination. And there are just lots of links between splicing and transcription. So this isn't really specific to polymerase poisoning, but it is specific to alternative transcription. We see PDCD4 inhibits translation initiation in mRNA, and lupus actually occurs again here. So some of these genes would be quite interesting to look at through the lens of lupus if you were interested in that disease. I'm not going to go over the end of this here, but I do think that these terms showed up quite interestingly. If we had more time, we could do a lot more natural language processing analysis of these results. In the future, I would like to improve the literature search by doing tokenization and part-of-speech tagging. We can use lots of packages for figuring out what part of speech each word is in a sentence, And we can actually pull out sentences that are related to our clusters of genes, perhaps finding the most descriptive, top 10 sentences or something to give us a feel for what our clusters are. We can, of course, we can do synonym detection for combining terms, and really importantly, we should use ensemble mapping or entree mapping, which gives us the PubMed IDs for every single gene, which is a much better method than I used for of just doing searches for the gene names as they're written in the HG19 reference. It would be interesting to create a knowledge base. There are some algorithms for using natural language processing for creating knowledge databases from literature. This would be an interesting thing to try to do with these clusters. And we could use the clusters to analyze upstream region and downstream regions and try to figure out if we could predict which cluster a gene would belong to by analyzing those regions. That would involve feature elimination and supervised learning. If we were successful in doing that, we might be able to identify genetic regulation or alternative transcription. And we know that there are some motifs in genes that provide for alternative splicing or alternative transcription. So we could expect to have some success in this approach. In the short term, I should contact Seqmonk and see if they would like to add the new code that I wrote. Okay, thank you for listening to my third rotation.